ISK is developing a novel anti-cancer immune therapy. ISK technology is based on the developing of a novel drug named Nerofe. Nerofe has the capabilities to activate anti-cancer immune cells specifically against cancer cells. It can also induce direct death in the cancer cells. So basically, there is a combination by both a direct apoptotic effect on cancer cells that overexpress Nerofe's receptor, and also activation of the innate immune response cells against cancer cells. The mechanism of action of Nerofe is based on the interaction between Nerofe and the immune inhibitory checkpoint named ST2. Originally, ST2 was discovered in 1995 by a group of scientists, which they took 33 immortalized cells and transformed them with mutated KRAS plasmid. The outcome was an overexpression of a novel protein which was named at that time by the name of T1. Later on, when the human parallel was discovered, they named it ST2. So the human ST2 has at least 3 splice forms. 2 of them are expressed over the cell membrane, and 1 of them is overexpressed in the Golgi apparatus. The splice format that we are interested in is the one that resides in the Golgi apparatus. In this slide, we can see cells, wild-type cells, that they overexpress ST2 in the Golgi apparatus in CT26 cells, which are murine metastatic colorectal cancer cells. As you can see on your right hand, the green stained cells, and on your left hand you can see the cells which are the knockout of ST2. They do not express, almost do not express ST2. When we applied Nerofe to these 2 cell types, the wild type responded nicely with a decrease in viability of cells in a dose-dependent manner, as opposed to the knockout, to the ST2 knockout cell line, which did not respond to Nerofe. So basically, Nerofe is working on cells by interaction with ST2. What is the mechanism of action? What is happening in cells after Nerofe interacts with ST2? As you can see in this slide, on the left side, we can see cells in which we applied Nerofe to it. Nerofe bounds to the extracellular ST2 receptor, then it comes into the cell and concentrates in the Golgi apparatus, where the soluble ST2 splice format is concentrated. Once Nerofe is binding to the Golgi apparatus, to the ST2 in the Golgi apparatus, it inducing oxygen free radicals, which cause complete destruction of the Golgi apparatus. Once the Golgi apparatus is destructed, then a process named ER stress is being evolved. Proteins which are translated in the ER have no way to go outside because there is no Golgi apparatus. and due to this heavy ER stress load, apoptosis is being induced in the cancer cells. Before we started our clinical trials and clinical work, we thought what might be the best combination of Nerofe that will increase the drug capabilities to kill cancer cells. We thought about different combinations, but it turns out that the best one is low dose doxorubicin. What is the advantages, or why we added Nerofe to doxorubicin or doxorubicin to Nerofe? In vitro experiments shows that when one apply Nerofe to innate immune cells like NK cells, DC cells, monocytes, there is a very strong activation of these cells. From the other hand, if one takes cancer cells and treat with doxorubicin, these cells become highly susceptible to NK and DC cells. So if we will combine Nerofe with doxorubicin together, we will be able to see that from one side there is an activation of the immune cells, NK cells, DC cells, and from the other side the cancer cells are becoming very sensitive to NK cells and DC cells. So the combination has very strong anti-cancer capabilities. Now, one very interesting question is why or how the NK cells or dendritic cells will collaborate with, will see the cancer cells. Very interestingly, once, as I said before, Nerofe is being applied to the cells, there is induction of severe ER stress. It is well established that once severe ER stress is evolved, there is an interesting protein molecule named CRT. CRT is a chaperone that resides in the ER. When there is a severe induction of ER stress, CRT is moving from the ER to the cell membrane. CRT expression over the cell membrane serves as an eat me signal for NK cells and dendritic cells. So basically, from one side, Nerofe activates the NK cells, the dendritic cells. From the other side, the cancer cells are expressing the eat me signal for the NK cells and dendritic cells, and they are attracted to the cancer cells, which are now also highly sensitive to NK cells and dendritic cells because they were treated with doxorubicin. So this combination is a great combination. Second reason why we added Nerofe to doxorubicin, low dose doxorubicin. Nerofe induces a CHOP. CHOP is a protein that under ER stress conditions being strongly expressed in the cells. It turns out that cancer cells that express CHOP are highly sensitive to doxorubicin as well. So again, the combination has a very strong capability, very strong anti-cancer capabilities. Third, Nerofe is inducing ER stress in cancer cells. Doxorubicin has the ability to inhibit the ER stress repair mechanism. So once we introduce Nerofe plus doxorubicin, the severity level of the ER stress is increasing dramatically due to inhibition of the repair mechanism of the ER stress. It's well published, it's well known, it's no doxorubicin, I just said doxorubicin serves in low doses as an inhibitor for an enzyme that is responsible for splicing of one of the transcription factors that activate the repair mechanism of the ER stress. So these are at least 3 different reasons why we combine Nerofe with low dose doxorubicin. When we tested this combination in vitro, we were able to show that if one introduced Nerofe to some cancer cells, we can see a decrease in viability to 10%, 15%, in some cases 20%. Doxorubicin in low doses, the same, 10 to 15%. When you combine them, we can get between 50 to 70 direct apoptosis effects. Increasing in production of oxygen radicals. When you apply Nerofe to cells, you can see around 60% in increase of production of oxygen radicals. Doxorubicin is adding 10% together, we can get between 60 to 80. Mir-217, it's a microRNA that has very strong anti-oncogenic effects. So basically it turns out that Nerofe can increase the level of Mir-217 by 200% in some cells, in others 300, 400. Doxorubicin, low levels around 400%. If you combine them, we can get between 60 to 900%, in some cases 2000%. Decrease in expression of protein, KRAS/MAP kinase 1 and 2. Nerofe by itself can decrease the protein expression by 10%, doxorubicin by 25%. If you can combine them, again, you can get between 90 to 100%, and we think that the reason for the severe decrease in the expression of KRAS is due to the high expression of Mir-217, which has 2 binding sites in the RNA of a KRAS in a 5' untranslated and 3' untranslated. So these are more or less theoretical considerations with some preliminary results. When we looked at cell viability, and we concentrated on 3 cell lines, 2 human cell lines, 1 murine cell line. The human cell lines, they are all metastatic colorectal cancer transformed by KRAS. As you can see, here is the results of applying Nerofe plus doxorubicin to human SW480, again metastatic colorectal cell line. Here is the level of the control viability of cells, 100%. Nerofe added some. Doxorubicin did not do a lot by itself, but when we combine it with Nerofe plus doxorubicin, we were able to see like 70% decrease in cell viability. Same story with CT26. This is a murine colorectal cancer, a metastatic colorectal cancer cell line transformed by KRAS. You see here is the level of the untreated group of cells. Nerofe and DOX together like 20-23% decrease. When you combine them, we can get up to 85-88% decrease in cell viability. The same story with HT29. Okay, what about Mir-217? Mir-217, as I said before, is a microRNA that can decrease the level of expression of proto-oncogenes in cancer cells. So we looked at Mir-217, and as you can see, in SW480, you can see clearly there is a very strong increase with the treatment of Nerofe, doxorubicin, and the combination. You can see down here the quantitative analysis. This is U6 as a control of level of microRNA in the samples. Same story in HT29, same story CT26. As I said before, MIR217 can downregulate expression of the KRAS protein. So we asked whether we can see a real decrease in the protein levels of KRAS in these cells. So we looked at SW480. Here's the level of KRAS, untreated group, Nerofe, Doxo. Together there is almost complete elimination. CT26, the same story. HD29, the same story. You can see here the quantitation. Here is the level of KRAS in SW480. In the group of Nerofe, a decrease with DOX, almost complete decrease in the combination. Same story with CT26 and HD29. So basically the combination can downregulate the level of expression of KRAS. All of these experiments are in vitro experiments. What about in vivo experiments? So what we did, we took CT26, we inoculated them under the skin in biopsy mice, and as you can, and then we inoculated them under the skin, and we were looking after the tumors to start to grow. When they start to grow, we divided the mice into different groups randomly, and we started the treatment. You can see the graph describing the tumor volume of the control group. At this point, we needed to take all mice out of the experiment. In the group of Nerofe and DOX, each one by himself, there was a prolonged in the capability of the mice to stay in the experiment for about, I would say, like almost 10 days. But again, there was an increase in the tumor volume, and then we need to take them out. In the group of the treated with the combination, you can see very nicely there is some increase, and then there is a plateau here. So the tumors are not as big as with DOX and Nerofe. And what is interesting, when we send the tumors to pathology, the report we got back is that in the group of Nerofe and DOX, most of the tumor inside the core of the tumor was also almost empty. Cells were dead there. And we were able to see a lot of immune cells around the tumor, and I'll show you pictures in a minute. After that experiment, we sent the tumors to pathology, and then we got back the biopsy. And the first thing we did is we stand for a KRAS expression and stand the samples with DAB. As you can see here is the control treated with Nerofe DOX, and together there is almost no expression of KRAS. What about the microenvironment? Very interestingly, when we stand the biopsy for anti-interleukin 2, you can see that in the untreated, treated with Nerofe, DOX, treated with DOX, there is almost no interleukin 2 expression. Look what happened in the tumor when there is a combination of Nerofe and DOX. Same story happened when we looked at cells that are expressing interferon gamma. In the untreated group, there is no cells that are expressing interferon gamma. Same thing for Nerofe DOX, but when we combine them, we were able to see very nicely expression of interferon gamma. Presence of NK cells. NK cells, we were not able to detect them in the untreated in the Nerofe and DOX, but when we combine them, we were able to see a lot of NK cells in the tumor. Same thing for M1 monocytes. You can see here when these cells are double stand for CD68 and interferon gamma, and they look like yellow. So these are M1 monocytes. So basically what we see here is that the cells, the tumors that were treated in mice that were treated with Nerofe and Doxorubicin, there is a complete change in the microenvironment as opposed to Nerofe by itself and DOX by itself. To finish this presentation, I want to give you some taste of the clinical capability of this combination. Here is one example where we took a lung cancer patient, stage 4. She was positive for ST2. This lady was suffering very badly, and then she was treated in a clinic in Zurich and Switzerland, and then she was sent home because no treatment was available for her any longer. As you can see here, when she started, this is the picture of the metastasis in her body. This is what happened after 4 months of treatment. You can see here it's a different perspective. You can see this is on day 1 of treatment. This is after 4 months. So I'm very pleased to show you that going from the preclinical work to the clinical work that we are doing, we are very excited about this novel immunotherapy. We are now in phase 2A trial in Miami looking at AML patients. We are doing a phase 1B at the Georgetown University at Washington DC looking at mutated KRAS tumors, and hopefully we will be able to help humanity. Thank you very much for your time.